For this video, I'm going to cover topic 16, inherited change, and I'm going to focus on a bit of process of meiosis, but my main thing is to show the difference between mitosis and meiosis, because I know that a lot of students, they can just memorize the whole process of meiosis, but they couldn't understand why the process of meiosis is going to result in reduction, division, where the chromosome numbers actually reduce from 2n to n. So I'm going to help you to clarify the doubt. This question from May June 11, paper 43, is a very good overview to show your understanding if you can actually solve this question. We have meiosis 1, which is divided into 4 stages known as prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2, likewise, you're going to have prophase 2, metaphase 2, anaphase 2, and telophase 2. So we're going to match these stages. So how do we identify prophase 1? As we know that during prophase 1, homologous chromosome will actually pair up. So we're going to start with B, where you can see that chromosomes exist in a pair, where you can see 2 long chromosomes and 2 short ones. And homologous chromosomes are actually having same genes at the same locus. So during prophase 1, which is the longest phase in meiosis, is because it involves process of pairing up of homologous chromosome known as synapsis and crossing over, where there's exchange of allele between the non-sister chromatids of homologous chromosome, which is indicated here between the black color chromatids and the white color chromatids. So this is where you're going to see a new combinations of allele. So this will be step 2 for E, and step 3 will be crossing over, which is still part of prophase 1. So after they are done exchanging the allele, the spindle fiber will pull and arrange the chromosomes to the equator. This will be step 4, metaphase 1. So as you can see that the chromosomes are still in a state of crossing over, where the process continues. And once they are done, as spindle fiber starts to contract and shorten, you will see that the homologous chromosomes actually gets separated and individual chromosomes move to opposite poles. As the chromosome reach the poles, the chromosome will start to uncoil and they will become chromatin again. So, and at this point, you will see that the chromosome is already reduced in number. And based on diagram F, you will see that 2 of the white one will be moving to one pole. So this is known as a haploid nucleus, because instead of having 4 chromosomes, each of them actually have 2 chromosomes remaining. And as you can see, the chromatids of each chromosome is already no longer identical. So this is when haploid nucleus or haploid cell is already formed. And this is step 6, telophase 1. So that is the end of meiosis 1. Meiosis 2, you will realize that the process is similar to mitosis with some slight difference, where you can see that they started off with non-identical sister chromatids. This is prophase 2. So you can see the sister chromatids are slightly different. And the next step should be metaphase. So now the sister chromatids that looks non-identical will be arranging at the equator. This will be step 8. You will have metaphase 2. Can you see the difference between the H and I? For the H is the homologous chromosome lining up at the equator, but for metaphase 2 is just the chromosome lining up at the equator. And after metaphase, we will have anaphase. So for anaphase, you will see that it's the sister chromatids that are being pulled to opposite pole. And for telophase, you will see that you have another 2 nucleus form, but now the nucleus is still a haploid. So take note, they don't get reduced again, because if you look at the number of chromosomes, there are still 2 chromosomes in each cell. The difference is just that the chromosomes are no longer with 2 sister chromatids, but each chromosomes are having only 1 chromatid, which is like the origin. So this is the last step, known as telophase 2. So you will realize that meiosis actually involves 2 cell division. Therefore, you will have 4 cells in total. In this diagram, they only show 2 because at the end of telophase 2, this is actually the outcome from one of the cell from meiosis 1. So if you times 2 of the cell from meiosis 1, you will get a total of 4 haploid cells. So let's have a quick overview about the difference between mitosis and meiosis. Let's look at the overview difference between mitosis and meiosis. First thing, the criteria is, we know that meiosis is meant to provide or to produce genetic variation in a cell, which we know that in mitosis the cells are identical, therefore no genetic variation, and meiosis actually have genetic variation. And number 2, we know that meiosis, the purpose is for gametes production, therefore you need a reduction division, where it means your chromosome number is reduced from 2n to n, which is not happening in mitosis because they want to maintain the diploid number, and therefore it's present in meiosis. So if we want to talk about where this takes place, cells that's actually involved, mitosis actually happens in all the somatic cells, which means all your body cells, excluding the gamete formation. But actually during the process of gamete formation, we do have mitosis, so that you can have millions of gametes formed, but we will focus on the somatic cells. And meiosis is only found in your gonad, which is your sex organ, which is during the production of gametes. So how many cell division does each process consist of? Remember, in mitosis, you just have one set of P, M, A, and T. So it's only one. For meiosis, so you have 2 cell division. That means you have twice the number of cytokinesis. So you end up with how many cells? This one you will get only 2 cells because one cell split into 2. While this one you have one cell splitting into 2 and 2 splitting another one more time. So you get 4. So this is actually the general overview between the difference of mitosis and meiosis. Now let's zoom into the detail. Let's think about the prophase. So during prophase 1, what makes the meiosis prophase 1 different? So in meiosis 1, there's crossing over, which is not existing in mitosis. Therefore, mitosis is producing genetically identical cell. And how is crossing over possible? Because there's a synapsis where homologous chromosome pairs up. So not present in mitosis, but present in meiosis. Now moving on to metaphase, let's look at the arrangement of chromosome at the equator. So during mitosis, we have chromosome lining up, whereas for meiosis, you have homologous chromosome lining up together. Remember, they are still in the midst of crossing over process. Therefore, both of the chromosomes are still pairing up next to each other. Then the separation of the chromosomes during anaphase. So how do they differ? As you can recall, since the chromosomes are actually lining up in pair for homologous chromosome in meiosis, therefore when they separate, is the pair that is being separated. Pulled by spindle fiber. In mitosis, is the chromatids that are actually separating. So at the end of telophase 1, haploid nucleus is already formed, whereas at the end of telophase of mitosis, a diploid number of chromosomes remain, as the chromosome still has pair in each of the cell, whereas in meiosis, the pair of chromosome has already separated. Hope this reinforces your understanding about meiosis, which if question asks you to make comparison with mitosis, and you are capable of handling it. If you have any other doubts, do not hesitate to contact me for group or private tuition. Thank you.
That's all for today's video. If you are interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.